Good morning. Today we'll talk about DNA. The DNA is made up of a double helix structure composed of two complementary filaments of nucleotides. At every nucleotides corresponds one of the four bases of the genetic code: thymine, adenine, cytosine and guanine. But we'll talk about DNA aging. DNA aging is characterized by three factors: epigenetic alterations, telomeres shortening and instability of the genome. Epigenetic studies the chemical alterations of the DNA that change the gene's expression without altering the basis sequence. For example, some molecules can attach to the mentioned elements as we see in the picture. It was noticed that these types of molecules are frequent in the elders. That's why they were thought to be a part of the aging. The genome is unstable, the DNA filament can be damaged and not be able to replicate again. The filament can break and be repaired by the ligase enzyme. There also can be the formation of the dimers. The telomeres protect the DNA from losing genetic information. As a matter of fact, they don't have any genetic information. However, some cells produce the telomerase, that rebuild the telomeres. It can be found in the tumor cells. That's why they are so strong. It was discovered that with the potassium cations the telomere assume a particular form of tetraeds stacked. Assume this form, the telomere doesn't allow the telomerase to do its work. That's why the phenomenon is so interesting, since it could be useful against the proliferation of the tumor cells.